Hi guys, let's continue with our component of the plant cells. As I mentioned earlier, we have cell wall as the outermost layer and everything inside is protoplasm. And later you learn about the word protoplast is just a unit of protoplasm. We can count 1, 2, 3, 4 for protoplast, but protoplasm is more like a description of everything inside the cell wall. And what are inside the cell wall? We have learned about the plasma membrane just a little bit, and inside the plasma membrane, you have organelles. And we already know what are organelles. It's just the living part with the biochemical activity. And we learn a little bit about the non-living parts, which is the inclusion. So here I have some notes that cytoplasm is equal to protoplasm except plasma membrane, nucleus, and vacuole. You later you might need to heard about cytoplasm. So let's come back to organelles. The example of organelles are nucleus, chloroplast, mitochondria, endoplasmic reticulum, Golgi apparatus, vacuole, and lipid droplets. For inclusion, examples are crystal and starch granules. So let's start with the first component, cell wall. The function of cell wall is to define the shape of the cells. Position in the cell surround the position of the cell wall is surrounding the plasma membrane. The component of this you can see right here is composed of cellulose, small like the yellow rods. Cellulose is the glucose molecules connected to each other from 100 to 15,000 molecules in a long chain. Cellulose provides strength for the plant cells. The next component is hemicellulose showing in the brown color right here. It acts like a glue-like substance that holds the cellulose together. Hemicellulose is another type of carbohydrate. It's more like several substances combined together. The last one is pectin, showing in the deep blue colors. These are the complex polysaccharides that allow the plant cell wall to extend. It can expand and grow larger in size. All right, the plant cells provide strength and it's around the plasma membrane. The plasma membrane can be seen in the bottom of this picture. All right. So there are two types of cell wall, the primary cell wall and the secondary cell wall. In general, primary cell wall is thinner, it's more flexible, it is present in all the plant cells. Every type of plant cells, you need to have the primary cell wall. It consists of cellulose, hemicellulose, pectin, and glycoprotein, which I do not mention earlier. It's just the combination of protein and sugar together. And primary cell wall located in the outermost layer of the cells. The secondary cell wall built inside the primary cell wall. So the secondary cell wall is more rigid and it can be found in some type of cells, not all the cells, unlike the primary cell wall. It produced inside the primary cell wall and is derived from the primary cell wall by the thickening of the primary cell wall and in addition with lignin, which provide the strength to the secondary cell wall. As I mentioned earlier, it can be found in just some type of cells that need the strength. For example, in the xylem of the plants, it need to stand from the very high pressure during the transpiration of the plants, or the cells of xylem is called vessel elements, and some fiber cells and some type of cells called sclerenchyma. We will learn about that in this lecture. I'm not sure which video you will learn about sclerenchyma. And secondary cell wall contain more cellulose than primary cell wall. So um, between the cells, plant cell need to stick together, and cell wall by cell wall it can be cemented or glued with the substance called middle lamella. This is the layer of pectin that act like a glue, and this part of the plant is too thin to be visible by the light microscope. In this picture, you can see that middle lamella is present in green color, and the red one right here is primary cell wall. The next inside is secondary cell wall, and then you have plasma membrane and cytoplasm. There are another feature called plasmodesmata. It's just a passage between the plant cells. So this is the cross section of the maize or corn stem. You can see that I forget to mention. So the primary cell wall is more flexible and the secondary cell wall is more rigid. By having the lignin in secondary cell wall, you allow the secondary cell wall to be stained with the red dye. But the primary cell wall do not have lignin and it cannot be stained with the red dye. As if you can see right here, this one is the vascular bundle of the maize stem. The red color cells are composed of secondary cell wall, while the cells without the red color, the green or the blue one, are primary cell wall only. All right. So this is another extreme example. Scarid or sclerenchyma cells or stone cells. This one is from a pear fruit, and this one is from the leaf of some tree. These two type of cells are scarid. It has a very thick secondary cell wall. Inside the thick secondary cell wall, you have a space. It used to be cytoplasm, and after the cell die, this cavity become a lumen. And once the cell is still alive, there are passage between inside the cell and outside. This passage is called pit canal. All right. The next component I want you to learn about is plasma membrane. Plasma membrane is very important because it defines the space, and all the cells need to have plasma membrane. It's a definition of the cells that need to have the membrane. It's um, it's the boundary between extracellular space or outside and inside. If you notice, outer outside of the cell and inside of the cells are a substance that like water. We call this substance as hydrophilic substances. And if you have to separate between the two, you need to have something that do not dissolve in hydrophilic environment. Excuse me. So the plasma membrane has that properties. If you take a look at one unit of lipid bilayer, this one is called lipid bilayer. Each unit of it contains hydrophilic head. This part can dissolve in liquid, and The half other part is hydrophobic tail. This part can be dissolved only in oil but not in water. If you put this back into the lipid bilayer structure, you can see that the outer part, the hydrophobic head, this one can be um, be next to the hydrophilic environment outside of the cells. Likewise, this hydrophilic head of the lipids can be next to the hydrophilic environment in the cells. And in the middle, you have the hydrophobic tails that can act as a supporting draft for the membrane to hold everything together, and it needs to be non-soluble in hydrophilic environment. So the major components of plasma membrane is the lipid bilayer. Everything else like proteins or carbohydrate is just the addition. And right here, I want you to take a look at the plasma membrane, which is a unit of, which is protoplast, which is a unit of protoplasm within the plant cells. We can separate the protoplast by adding salt or sucrose for the plants, so the plant cells will lose water. The cell will become, the protoplast will shrink, and then you can see the separation of protoplast from the cell wall. Like in this microscope, these are the cell wall, and the purple substance in the middle are protoplast that's separating from the cell wall. The next component is chloroplast. Chloroplast is the machine, it's act like a machine to convert the sunlight and use that energy to assimilate carbon to produce sugar and then starch later. This is the structure of the chloroplast. You don't need to go into the detail at this point. Later, you will learn about the structure of chloroplast and the function of the chloroplast in the photosynthesis lecture. Right here, I just want you to know that chloroplast has different shapes in different kinds of plants, like moss. This is just one cell of the moss, and inside you see circle green colors. Those are mosses chloroplasts, but in some algae, their chloroplast has different shapes, like in Spirogyra, their chloroplast is like a spring or spiral shape, like this picture. We'll get more detail in the lab later. The next component is nucleus. Nucleus is just a very important organelle that contains genetics information or DNA in the chromosome. So it is the site of chromosome replication, and it is responsible for regulating metabolism, growth, and cell differentiation. It is surrounded by a membrane It's called nuclear envelope, and it's contain nuclear pore that allow the DNA to allow RNA to the products of DNA to leave the nucleus. All right. So if we talk about nucleus, we need to learn about cell cycles. Cell cycle is just the cycle of the cells that can divide in the mitosis part and non-dividing in the interphase part. During the interphase, the cell prepare itself to be divided. Something like the cell increase in size, produce the ribosome, RNA, and substances involved in DNA replication, organelles division, and production of microtubules and some other components that important for mitosis. Mitosis is the phase of cell division. It's composed of prophase, metaphase, anaphase, and telophase. We'll go into detail later. And after the cell division, there is a cytokinesis. Whatever is remain undivided, it will be divided in this phase of the cell cycles. So let's get into the prophase, which is the first stage of mitosis. So during the interphase, the DNA is just cannot be visible because the chromosome is relaxed. Everything is sparse and you cannot see any DNA. During the prophase, chromosome become condensed and shorter and thicker, so you'll be able to see some coil of chromosome, and then nuclear envelopes start to dissociate from the nucleus, and nucleolus just disintegrate or disappear. And then in the metaphase, chromosome become very dense and align in the middle of the cells. In the anaphase, the two chromatids of one chromosome is separated into different opposite direction or opposite pole of the cells. The last phase of mitosis is telophase. Each group of daughter chromosome become surrounded and reform. The nuclear envelope just become reforming again. The daughter chromosome, or in the last slide we call it sister chromatid. Later, each sister chromatid double themselves to become a new chromosome. We call that daughter chromosome, and it will become longer, thinner, and finally indistinguishable. They are tangling together, tangling around each other. Nucleolus become reappear, and many mitotic spindle disintegrated, and the cell plate right here is formed again. All right, we finished with the component of the organelles and also the cell cycle. The next component of the cell is cell inclusion, which is the non-living substance that are not be able to carry out any metabolic activity and are not bound by membranes. Example of that are calcium oxalate crystal. You see this crystal in the lab, and the other one is starch granules. This one become purple because it's is staining with iodine and then become purple. Otherwise, you see the clear structure, which is difficult to see. In addition, cell inclusion example are pigments and some secretory products like resin and tannin. All right, we finished the component of the cells, and in the next video, I will talk about the type of the plant cells and tissues. All right, thank you very much, you guys, and see you in the next video.